What is Wilson's disease? It is a genetic disease caused by mutation, random occurrence that alters an organism's DNA code. These alterations can sometimes benefit an organism, which explains how they evolve and adapt to an ever-changing environment. However, as much as mutation might benefit an organism, it can also provide negative effects, such as Wilson's disease. Wilson's disease, or hepatocellular degeneration, prevents the body from removing extra copper. Even though our body needs a small amount of copper from food to keep us healthy, too much of it will be fatal. Let's take a look at how our body processes copper. If there are excess amounts of copper in the body, the liver filters extra copper and releases it into bile. Bile is a fluid that is considered as a toxin or waste, thus is excreted out of the body through the gastrointestinal tract. The gastrointestinal tract consists of the esophagus, the stomach, the small intestine, and the large intestine. Therefore, copper is excreted out of feces. In Wilson's disease, the liver does not filter copper correctly, as there is an error in converting copper into bile. Therefore, excess amounts of copper builds up in the liver, brain, eyes, and other organs. High copper levels in the body cause organ damage, resulting in fatal symptoms. Wilson's disease was first discovered in 1912 by Samuel Alexander Kiner Wilson, who discovered this disease while diagnosing a patient who had cirrhosis, a disease caused by liver failure and neural disorders. He realized that the symptom was potentially inherited, since the patient's family bloodline all had similar symptoms as well. Then how does Wilson's disease occur? We first have to explore a simple genetic principle that follows when offsprings inherit traits from their parents. According to the general principles of genetics, a trait is an attribute that is physically expressed on an organism. Examples include hair or eye color. A trait is composed of alleles. Alleles are alternative forms of a gene that make up a single trait. A trait is always composed of at least two alleles. There are two types of alleles, dominant and recessive. Dominant alleles always are expressed over the recessive ones. When genes are inherited, each allele from the parents combine randomly to form the genetic makeup of an offspring. Since combining randomly occurs, we can determine the probability where the offspring will inherit a certain combination of genetic makeup. This probability can be determined through a Punnett square, where it shows all the possible combinations of genes that an offspring will inherit from its parent. For an offspring to express a recessive trait, one must inherit both of the recessive alleles from both parents. Wilson's disease is genetically recessive, meaning that a child has to inherit both of the parents' recessive genes in order to get the disease. This type of inheritance is also known as an inherited autosomal recessive mutation, where genetically recessive trait is mutated and propagated through generations. There still lies the question on how Wilson's disease occurs in the first place. First, we have to take a look at the chromosomes. Chromosomes are DNA strands that are tightly packed together. Each region in a chromosome encodes for different traits in an organism. When babies inherit traits from the parents, they essentially inherit 16 chromosomes from each parent's side. Therefore, every human body will always have a total of 32 chromosomes in its genetic makeup, and so are their parents. Wilson's disease is expressed when a mutation occurs in the ATP7B gene, which is located within the Q14.3 codon on chromosome 13. The ATP7B gene plays a major role on making the body possible to remove excess copper that it digests. The mutation on that gene prevents the body to successfully convert copper in the form of bile, leaving excess copper to build up within the body. Then what would be the probability of offsprings inheriting Wilson's disease? Let's look at the Punnett square again. Since Wilson's disease is a recessive trait, if we look at the probability distribution of the inheritance of Wilson's disease, the probability will be 1/4, or 25%, when both parents are carriers. Therefore, Wilson's disease is not a rare disorder. Approximately 1 out of 30,000 people express Wilson's disease globally. Since Wilson's disease is not carried by sex chromosomes, men and women both have an equal probability for them to inherit Wilson's disease. In Korea, 1 out of 100 people express Wilson's disease. Wilson's disease expressed in ages range from 3 to 73. Therefore, there is no specific population in which are the most affected by this disorder. It is evenly spread out. Since copper buildup affects primarily on the liver, symptoms of Wilson's disease are analogous to a person who has liver failure. These symptoms include weakness, fatigue, loss of appetite, nausea, vomiting, and muscle cramps. Also, copper buildup within the body forms a yellow copper ring on the iris of a person's eye. This ring is also known as a Kaiser-Fleischer ring. The symptom is the most unique that stands out as Wilson's disease. High copper concentration also affects the central nervous system, causing symptoms such as tremors, muscle cramps, changes in personality, depression, and anxiety. Now that we know pretty much about Wilson's disease, how can it be diagnosed? The first approach is to check whether a family member had Wilson's disease. If they have, then there is a high chance that a family's generation will have the disease also. Another approach would be to check if a person has unusually high copper concentration or low ceruloplasmin levels. For most people who have Wilson's disease, an enzyme called ceruloplasmin lack within their body. Ceruloplasmin is a protein that transports copper to excrete them out of the body. By checking the existence of a Kaiser-Fleischer ring on a person's eye, checking pulse rates, blood or urine sample tests, MRI and CT scans, one can calculate the concentration of copper and ceruloplasmin levels within one's body to accurately diagnose Wilson's disease. Further tests to diagnose Wilson's disease will be to perform a liver biopsy. A liver biopsy is a process of extracting a small amount of liver tissue from the patient. The tissue is then observed to determine diseases occurring within the liver. Although there are many methods to diagnose Wilson's disease, unfortunately, the disease cannot be prevented. However, one can expect that they might have Wilson's disease by checking whether their family member ever had Wilson's disease before. Again, Wilson's disease is inherited through bloodline. Then what are the treatments for people who have Wilson's disease? Wilson's disease cannot be cured since we don't yet have the technology to alter our genetic makeup. Instead, a major approach towards treating Wilson's disease is to manually remove excess copper from the body through medication and reduce the amount of copper entering the body through controlling one's diet. The most effective medicine used to remove excess copper from the body is through taking chelating agents. These agents are known to be better than D-penicillamine, an original medication used to remove copper. Chelating agents react in the body to release excess copper built in organs into the bloodstream, where they can be further processed by the kidneys and excreted through urine. A major side effect towards taking chelating agents are that it causes further neural damage, worsening one of the symptoms caused by Wilson's disease. Therefore, additional medications, such as titrine, is prescribed to hasten neural recovery and plasticity. Zinc is then prescribed to patients, since when the body absorbs zinc, it does not absorb copper to enter within the body. Patients who have Wilson's disease should be careful with their diet. They should not eat foods that are high in copper concentration, such as shellfish, liver, mushrooms, nuts, and chocolate. They should also be careful with the water that they drink. If their drinking water has high copper concentration, patients with Wilson's disease should avoid that and drink water with minimal copper concentration. As you know by now, Wilson's disease occurs from mutation, and this disease can be fatal to a person's life if not treated properly. So, are there only negative effects from mutation? Well, no. Mutations can also be beneficial. Mutations contribute to the factor of diversity within organisms of the same species. These diverse factors within organisms can result a species to develop immunity over fatal influences. Well, for example, by mutation, some humans are immune to HIV, a virus that causes AIDS. Some people carry a genetic mutation that makes them immune to HIV infection. It can be said that this beneficial mutation can propagate to larger spans of the human population, making the next generation of the human race to be immune to this harmful virus. Therefore, mutation contributes to diversity on a certain species. Every organism has their uniqueness. HIV immunity is also clear evidence that evolution still takes place in humans as well, thanks to mutation. Another example of beneficial mutation can be found in GMOs. GMO stands for a genetically modified organism. It depicts any organism whose genetic material have been altered using genetic engineering techniques. This process is called gene editing and is also considered as an intentional mutation. GMOs are used to produce various medications and genetically modified supplements and are widely used in scientific research, agriculture, and goods and services. The benefits resulted through the production of GMOs are most exemplified in food crops. Genetically modified food crops have resistance to certain pests and disease, making them healthy and long-lasting. They remain fresh longer than non-GMOs and are also higher on nutrient profile. The focus towards GMOs are this: they are superior than their non-mutated species in many ways. Therefore, mutation also contributes towards progressional evolution to a certain species. Benefit or not, the process of mutation guarantees one certainty: change. It is true that these changes are random, but it also gives us insight that Mother Nature is unbiased. Perhaps mutation is nature's ultimate wisdom: to constantly change, to think out of the box, and always be open to any opportunities of search space. Perhaps Wilson's disease, and like any other genetic disease, is a byproduct of nature's risks, all made for the hopes of a better cause, everlasting progress, and evolution.